We have three talks today, all by different participants in the center. So our first talk is from Cassie Avila. Cassie's going to talk about cholinergic circuits modulating interhemispheric communications. So Cassie is kind of a Udall veteran. She actually did her undergraduate work at University of Texas in El Paso, which I think is her hometown, if I remember correctly. And then was a graduate student here with Martin Sarter, where she did some really beautiful work on striatal cholinergic neurotransmission, and is now postdoc with Omar Ahmed, having migrated up to the cortex, in this case, the retrosplenial cortex. So Cassie, do you want to share your screen and get started? You're still muted, Cassie.
Okay, there we go. Let me go ahead and share my screen. 1 second. And beginning. And okay, everything look good on your end? All right, perfect. Thank you. Okay, well, good afternoon, everyone. Today I'll be discussing some of my research on how cholinergic inputs influence retrosplenial cortical function and interhemispheric dynamics during navigation, with these implications on how cholinergic dysregulation may contribute to spatial deficits observed in neurodegenerative diseases such as Parkinson's and Alzheimer's. So let's scoot that there. Okay. So spatial disorientation is a debilitating symptom that many Parkinson's disease patients struggle with, where they struggle to link external landmark cues to their internal sense of orientation. Notably, these deficits persist despite dopamine replacement therapy, which indicates that there's non-dopaminergic mechanisms at play. Similar spatial disorientation emerges early in Alzheimer's disease. Patients often lose their ability to navigate even in familiar spaces, which is commonly referred to as wandering. And understanding the neurobiological basis for both of these diseases could provide insight into therapeutic strategies. And notably, damage to the retrosplenial cortex, which I'll be discussing, results in spatial disorientation. Here, a patient with a cerebral hemorrhage in the left retrosplenial cortex attempted to draw. I'm going to get my pointer here. Okay, cool. Attempted to draw their route home, resulting in this disorganized map, which you can really see in stark contrast to their drawing by their son. And importantly, this isn't a memory issue. The patient was able to recognize landmarks and point them out on a map, but they couldn't orient themselves. And these similar spatial impairments are observed in both Parkinson's and Alzheimer's, this suggests that there might be a shared underlying mechanism. But what do we know about the retrosplenial cortex? The retrosplenial cortex is a key cortical recipient of process vestibular information, which is essential for navigation and spatial learning. It integrates egocentric information such as self-reference cues, like knowing where you are in space and your body, position, and movement, with allocentric information such as external cues like landmarks. These processes rely on reciprocal connections with regions such as the hippocampus and thalamus for spatial information, as well as inputs from the visual and cingulate cortices. And the question that arises from here is, how does the retrosplenial cortex communicate with other brain regions for successful navigation? And prior work in the lab looked at this through simultaneous large-scale recordings across regions and identified a unique rhythm during REM sleep, visible in the raw P, which I'm showing here, but I'm going to zoom into individual cycles so we can see this. And when looking at these plots over here, we're referring to different theta cycles, and specifically we're looking at the theta phases. So this 0 to 180 refers to the rising phase of theta, 180 to 360 to the falling phase. And when we look at this, looking specifically at the retrospinal cortex, you can see high frequency oscillations ranging from 110 to 160, which are precisely coupled to the peak of theta, even more evident on the right side here when we zoom in. And the lab has since termed these oscillations as splines due to their resemblance to the teeth of mechanical gears. And from there, the question became, who are splines talking to? Using bilateral probes, they found that splines exhibit this unique anti-phase communication across the hemispheres, where the ipsilateral side, for say example in this case, the left side, and the contralateral side, we'll just say the right side here, alternate their peaks and troughs. So they're precisely antiphase in this way. So incidentally, this nomenclature of splines became even more fitting, as you can see the resemblance to the mechanical interlocking mechanical gears. And relating this back to spatial navigation, the question became, how does navigation at different speeds impact splines? Well, just briefly here, you can see in the results, as the running speed of the animal increases, so does the spline theta coupling, and splines become even more precisely antiphase, suggesting that splines dynamically adapt in response to spatial demands to support navigation. The question then became, how are they generated? Well, splines are prominent in active brain states such as REM sleep and navigation, especially at those higher speeds. And during these states, acetylcholine release significantly increases. And looking at retrograde labeling of cholinergic neurons that project to the retrospinal cortex indicates that they originate from the basal forebrain. So potentially these terminals might be modulating terminals onto the thalamic neurons that before that there is any spatial processing in the retrospinal cortex. And that's precisely what the questions have been for my projects, is how do cholinergic inputs influence splines? One notable finding from previous work in the lab was that in an Alzheimer's rodent model, 5X-FAD, these mice show diminished cholinergic terminals in the retrospinal cortex, which you can see here in this frontal section, zoomed in even better over here on the left side, with showing these different layers of the retrospinal cortex. And you can see there's a marked decrease in the fluorescence of the fluorescence in the stain with this vesicular antibody. And the consequences of diminished cholinergic terminals was investigated by directly infusing saccharin into the retrospinal cortex to destroy cholinergic inputs. And as you can see, both spline power and the ratio of splines to theta is diminished on day 21 as compared to day 5 post-infusion. Therefore, my hypothesis here has been that dysregulated afferent cholinergic signaling to the retrospinal cortex contributes to spatial navigation deficits as seen in Parkinson's and Alzheimer's. So to quantify cholinergic signaling, we employed Brad sensors, which was developed by the Lee Lab. Briefly, these sensors are based on a modified muscarinic receptor, acetylcholine receptor, fused with a circularly permutated GFP, which enables real-time measurement of acetylcholine. And in addition to 5X and wild-type mice, I suggested to employ INIV mice, which model impaired choline transport, so we can really dig into how cholinergic dysfunction might impact spatial orientation and splines. And to do this, we're utilizing a TDT fiber photometry system. Our setup includes a commutator, and in these experiments, we also have the demodulated data filtered with a 6 Hertz low pass frequency to just minimize any high frequency noise. During the recordings themselves, the mice explore this open field so we can look at dynamics during natural movements, and we can look at the baseline walls as well as fluctuations to specific behaviors such as movement, initiation, and changes in speed. We extract this with LabGIM. I'm not going to get too much in the specifics, but essentially we can look at these different types of behaviors that we want to quantify. These are some of the examples here. And we can get to these different measurements such as time points for behaviors, counts, duration, things like that. And these are very preliminary results, but I just wanted to orient you here. So I'm presenting Z-scored average traces of the fluorescence changes corresponding during movement initiation as well as grooming. For these plots, I subtracted the isostoic signal from our GFP signal, calculated delta F over F for each value, and then extracted data points from specific time points, as I said over here. And then these resulting values were transformed into z-scores to standardize across animals. These are very preliminary results from 2 animals, but first what we can see is that there's this robust increase in acetylcholine levels immediately preceding movement onset and peaking around when movement begins. So this might suggest a potential preparatory role of cholinergic signaling in coordinating retrospinal activity for spatial navigation and movement. So the next steps will be looking at this a little bit further, such as like looking if there's changes due to running speed, mirroring what we saw with spines, and also investigating this in the INIV and 5X maps, just to really look into disruptions due to any deficits in movement precision or spatial navigation. And I'm going to skip a couple slides in the interest of time, but you can just so you can briefly see, I have some other fun stuff going on. But to dig into what our further experiments are, we'll be looking at the same kind of choline imaging in these animals, but also while they're undergoing different tasks that rely on allocentric navigation. And then there's a switch into relying on egocentric navigation to really see if there is changes in acetylcholine release when they're not switching these navigation strategies, and if cholinergic dysfunction disrupts the ability to do so. And this is important because this might reflect real-life changes faced by patients with neurodegenerative diseases, such as the difficulty to integrate these information is seen in Parkinson's and Alzheimer's. And ultimately, the experiments are aiming to provide insight into how cholinergic signaling supports spatial navigation and how disruptions in the system may reveal new strategies that we can utilize to mitigate the spatial disorientation. And with that, I'll just put up my acknowledgement slide over here. I want to thank the lab, as well as many of the members that have been in our lab, as well as the alumni, my undergrads, my T32 committee, Mark Bradshaw, who helped build a lot of these rigs, all the mice, and my funding sources. And I will take any questions at this time.
Okay, very good. Any questions for Cassie? So, Cassie, are you also doing any recordings to look at the spline frequency simultaneously? Yeah.
Not simultaneously yet. That is like the future aim, but now you gave me a chance to show off the rig, so thank you. So the other experiments I'm conducting are with neuropixel recordings so that we can look precisely what you're talking about here. The rig is set up over here. So in this specific instance, the animals are on this, they're mounted on this treadmill that rotates at different velocities. So we can at least answer some of those questions like, is our splines going to be modulated by these different frequencies? And this very brief example over here is just some like preliminary results looking at wild type versus 5X fad mice. And it's interesting because we see the same kind of theta coupling strength at higher speeds with wild type mice, but when you look at the 5X fad, we're not seeing that same pattern.
Hey, Cassie, nice talk. Just out of curiosity, are you thinking of this as being, I mean, I know, by the Martin Starter doctrine, everything is a transient response now, but yeah, the IAD9Vs typically have more of a difficulty with the sustain. So I'm wondering, like, are you thinking about so I guess my guess would be that you might not see a big deficit for them on at least the movement onset, but are you thinking about tracking it going on, or basically what's your thinking there?
Yes. So I understand your question to a degree of like, it may not necessarily be this transient effects that, yes, as you said, by the Martin doctrine, but one of the things I really want to get into with the imaging is to look more at different, one, quote unquote, bait states, but also I very briefly only got to mention that looking at the running speeds, just because we've seen that it is so highly prevalent with splines, and particularly as it strengthens. So as they run faster, so does the strengthening in terms of the coupling, but also with how precisely anti-phase they are. And one thing to think about is like, okay, if the idea is that they're modulating this cholinergic terminals before it can be processed into the retrospinal cortex. I would imagine it's going to be more on a large scale thing of like these brain states where you have high cholinergic tone. But if it's that these animals aren't able to even sustain that over a cross period of time as opposed to transient effects, it might be what you're suggesting more in the sense that it's going to be a longer, in terms of like a longer term duration.
Okay, great. It'll be fun to see how it works out.
Thanks, Cindy.
All right, very good. Well, thanks, Cassie. I think we're going to go on to our next speaker. So, next speaker is Julia Carli. Julia did her PhD work in Milan, and then was a postdoctoral fellow with our colleagues in Groningen, and now has come here to continue doing work on brain imaging in Parkinson's disease. Julia, you want to get started?
Yeah, sure. So let me share my screen. Can you see it? Okay. So thank you for the introduction. My work as a Udall postdoctoral fellow is right now focusing on understanding the heterogeneity of bidirectional changes in the brain of Parkinson's disease patients using neuroimaging. So the cholinergic system is very diffuse in the brain, cortically and subcortically, and this means that it can cause a plethora of different symptoms. Traditionally, cholinergic system is associated with cognitive impairment, but is surely not the only symptoms that can be associated with cholinergic system. And our lab has produced quite a fair amount of work to demonstrate that indeed cholinergic impairment are also associated with motor symptoms, sleep disorders like REM sleep behavior disorders, and degree of olfactory impairment and dynamic and gait and balance impairment. What is actually interesting is being the paradigm shift happening the last years, because cholinergic system degeneration was traditionally seen as a passive model. But what we are starting to notice is that indeed the cholinergic system can present some sign of what we think is a compensation mechanism. And here you can see two studies, one on the right side coming from our group showing that Parkinson's disease patient without cognitive deficit, they show an upregulation mechanism in terms of higher binding in the FLBV PET compared to healthy control. The FLBV PET is binding to the vesicular acetylcholine transporter, so it's showing the integrity of presynaptic cholinergic terminals. And on the left side, you can see that these upregulation mechanisms are present in cortex, thalamus, and various cortical regions, also in prodromal. And here specifically, the green box represent LRRK2 mutation carrier with prodromal PD, meaning that these compensatory mechanisms are beginning very early in the disease stage. Recently, our group has been working, and specifically Professor Bonan has produced this really interesting work showing that it is possible that to identify biological subtypes based on the amount of cholinergic upregulation and downregulation mechanism. Indeed, he found that there are some patients that present mostly only upregulation mechanism. Some of them, they present mainly downregulation mechanism, and other patients presenting a mix of these two changes. And this is important because these three groups also show different clinical symptoms. So meaning that these bidirectional changes we think they do have a functional meaning. What we don't know, and that's what I'm investigating, is that if the topography of these upregulation changes, so not only if they are upregulating, but also where is important and functionally important and able to identify groups of patients that might behave in the same way. And we are trying to do this at the voxel level approach in order to use upregulation and downregulation map in the future in the clinics to inform future therapeutic strategies. So I'm working with a multicentric data set in collaboration with the University of Groningen, where I did my previous postdoc. And we have Parkinson disease de novo, so without treatment, coming mostly from the University of Groningen, and patient with more mild advanced phase coming from the University of Michigan. And here you can see differences in the clinical and demographical feature. And the most important to take in mind is that, of course, the de novo patient, they have much less disease duration. Obviously, they don't have any levodopa treatment, but weirdly, they also have lower MOCA score, indicating a lower global cognitive functioning. And this is related to some selection bias. We think they are in place, and we can discuss if you are interested later. All these patients have been acquired with FOBV radiotracer. And what we are doing now is to create individual map for each patient, showing the downregulation and upregulation mechanism compared to a group of 30 healthy controls. So the type of map we are producing are exactly this one on the right side. In the blue scale, you can see an example of downregulation mechanism. So where patients have significantly lower uptake compared to healthy control, and in red scale, you can see the higher binding compared to healthy control that I will be calling upregulation. So with the collaboration with our data analyst, August van Hout, we are also applying some complex machine learning approach that is summarized in this slide. And the pipeline we are using is basically applying an approach that is called voxel-wide Gaussian mixture modeling, which what it does essentially is increase the dimensionality of each voxel, trying to enhance the topographical information of each voxel, creating a huge matrix of number that then we reduce with some data dimensionality reduction. For now, we use two kinds of dimensionality reduction, principal component and isomap. And then we apply a cluster approach to see if we can identify different groups. So these are examples of the heterogeneity I was referring to. These are patients from the University of Groningen, and you can see that the downregulation process can vary from none, so nothing, to various degree of downregulation in different regions. So, for example, this patient is more posteriorly affected, and this patient is more anteriorly affected. The same thing happened with upregulation mechanism, with patient not having any upregulation, other patients having a huge upregulation in whole the cortex or only selectively in the cerebellum, and so on. And the same is happening also in the University of Michigan cohort. As you can see here, various degrees of downregulation across the different patients, as well various degrees of upregulation across different patients. So this is the average of each image for each patient derived with the machine learning approach with the enhanced topographical information. And you can see on top the average of these models and then also the variance. On the average one, you can see that it's the region that are mostly upregulated are indeed the cerebellum, limbic region, their orbitofrontal cortex and prefrontal cortex and sensory motor cortices, and the downregulated region are usually more posteriorly parietal and temporal and occipital. But what is really interesting here is to look at the variance of this model, which is mostly fully overlapping with the upregulation mechanism, suggesting that that is where the patients varies the most, and that's probably what is going to help us to identify cluster. So with the principal component analysis, we selected 8 principal components because we wanted to at least explain 50% of the variance, but we also didn't want to include component explaining too little variance to not include noise. So that's why we came up with 8, but we are still exploring these options. And you can see here the silhouette score, and we decided to starting exploring. There are a couple of edge. There are arguments to make 3, 4, or 5 cluster, and we decided to start with 4 that are plotted here across the 2 components. And you can see that the groups are quite nicely divided. But if we use the isomap, a way to reduce the data, the dimensionality of the data, which is a different method compared to the PCA because it allows for no linear relationship, which is probably better for this kind of data. And we also go for 4 cluster. You can see that the groups are really much better separated. And that's why we decided to go ahead with the isomap results and 4 cluster. These are the 4 cluster of patients, and they really show very clear topographical distribution, with the first group having mostly only downregulation with a very selective upregulation in the cerebellum. The second group, which is composed mostly by the de novo patient from Groningen, is mostly upregulating without any downregulation. Third group, it's upregulating selectively in some region, some orbitofrontal and limbic region, and the fourth group is selectively upregulating in limbic region, plus some downregulation in the posterior cortices. And the interesting part is that these 2 groups, which are these 2, they show the best cognitive performance and also best motor scores, but with the same disease duration and same amount of levodopa of this group, meaning that these are probably subgroups that are not explained by the disease duration, the disease stage. And interestingly, these 2 groups are also younger. So probably the first group is kind of a severe phenotype of PD. But so to summarize, we can identify subtypes according to the topography, not only based on the amount of up or down regulation, and those subtypes seems to have a functional meaning. Also, the de novo PD patient, they might exhibit some kind of upregulation related to the dopaminergic system impairment that is not treated. So we are thinking to not consider the 2 groups together, but try to divide them because they are probably too different from each other, and they have different kind of upregulation mechanism. And the next step will be to explore different kind of classification approach, more complex than the k-means clustering, also incorporate longitudinal data to see how these subgroups are doing imaging-wise and clinical wise, and also we want to produce biomarker of expression of these different patterns in order to see if we can have a biomarker of disease progression to correlate with the longitudinal data and also clinical feature. So thanks to my mentors and to my group, also to the patients, and I'm really happy to take any question.
Thank you. So, Julie, so this used the levodopa dose as the measure of disease severity. Is that correct?
No, I used both. So I have disease. I mean, well, you mean I should include the ONR probably?
Well, I was thinking, did you use the DTBZ PET results as
No, we didn't covariate out anything.
Okay.
For now, we can do that. This is just an exploratory comparison across these groups to see if they actually differ in these variables.
Yeah.
And what I know right now is just that these groups are not different in terms of disease duration or neither in the amount of levodopa they are taking.
Right.
Of course, the second group is kind of a special group because it's mostly composed by de novo, and it's difficult to compare that group to the others. And we are kind of hoping with August because we think that if we divide these 2 cohorts, we might be able to grasp more subtle topographical changes that are more specific to the disease stage.
Right, right.
So this probably is not gonna stay like this. It's probably, I think it might be better to split them.
Yeah, but there'll be 3-year data from Groningen also, and then at that point they would look more like the Michigan patients demographically. So that would be, yeah, follow-up will be quite interesting. So
Yeah, we are looking forward to analyze them.
I find the cerebellum findings really cool. So to have this upregulation there in the cerebellum, this is very, very impressive. Does this correlate with any kind of clinical sign, let's say tremor or some other specific of the disease?
So the impression, so I cannot answer this question because I didn't try, but my impression is that this kind of upregulation is not functionally meaningful anymore, or at least it's not as functionally meaningful as the limbic one. So it might be that we will find some correlation, but I do feel this is something that it's more in late stages or maybe in more severe phenotype, and it might not be, I don't expect to find like huge clinical correlation, especially in patients that have a really severe cholinergic deficit all over the brain. But this is only my hypothesis. It might be that I will find something. I didn't try it, so like.
So Thomas, we do have some longitudinal data from our cohort, and there is at least an initial evaluation there is some difference in the magnitude of the cerebellar upregulation based on cognitive status. Right.
You mean Taylor paper? I have that slide.
Yeah.
So this I excluded this for the sake of time, but this is actually another interesting study we are about to. I mean, it's under revision, and indeed, as Roger was saying, is like these are only 2 groups, but what we did was to try to predict from baseline FLVV cognitive trajectories, and you can see here that patient that has persistent normal cognition, they do have upregulation, but mostly
frontal.
Yeah, mostly frontal. These patients that are converted MCI, they did have some upregulation, but in the cerebellum, and that's why I have that impression that maybe the cholinergic cerebellar upregulation, it's an effort to try, but I'm not sure it is a successful effort, let's say.
Thank you.
I think, Thomas, the other thing I'd say, I think in one of Nico's older papers, there is a relationship between some changes in the cerebellum and tremor. So yeah, it's a relevant question. So
yeah, we can see if in these subgroups maybe some motor subtypes are more common than others.
any other questions for Julia? Okay, let's go on to our third speaker then, Aaron Kucinski. Aaron's going to talk about optogenetic disruption of cholinergic mediated move control in rat models of Parkinson's disease falls. This is an extension of the prior work that he's done with the double lesion model that he and Martin Sartor developed. Aaron, do you want to go ahead and share your screen?
Yep. Where is it? Shoot. It's not working. Why it's not working?
Okay. Try.
I had it. We did it earlier. It worked.
Okay.
On my desktop.
Well, worst case, you could shoot it over to one of us and we could advance it for you. So.
Can you find the share button?
Yeah, no, the share button works, but it's not like earlier it worked, but it's not showing up when I hit. It's not showing up in the applications. Wait, hold on. Um, great. Yeah, I'm not sure what to do. It's not showing up. Sorry. When I
So
hit share,
your presentation has disappeared?
yeah, I mean, when we did this earlier about an hour ago, it showed up right in the bottom. It's not showing up.
Okay.
Can I send it to
Julia,
somebody?
did you unshare? Because if not, well, I guess no, because yours isn't on the screen anymore. I was wondering if maybe that captured it, but that's
You
probably
want
not.
to email it to me or to Nancy?
Yeah. Let me, yeah, I sent it to both you and Nancy.
Okay. Just wait for it to come through. Nancy, it might be best if I think you have the control if you control it. Wait
Okay.
for it. As
Yeah,
soon
I
as it,
think both of us should have, oh wait, here, now we see. Oh, there it
is that you,
is.
Aaron, is that you?
Yeah.
You got
Okay.
it.
Okay. Okay. I think it works now. Sorry about
Yes.
that. You
Yeah,
good?
that's great.
It looks fine now. Okay. Go ahead.
Sorry about that. Thank you, Roger. So the title of my talk is Optogenetic Disruption of Cholinergic Mediated Movement Control in Rats Models Parkinson's Disease Falls. So the data that I'm going to present today is kind of the third part of a series of experiments in which we've shown that silencing of cholinergic neurons of the basal forebrain causes, in part, Parkinson's-like movement impairments in rats. And we first done a lesion model, and then the second part was using chemogenetics or DREADS, and the final part of this trilogy of sorts is using optogenetics. So first, some quick background. Loss of dopamine in the striatum is, of course, the primary and traditionally defined deficit of the disease, and this is treated effectively with L-DOPA. However, there are L-DOPA-resistant symptoms such as poor posture, gait and balance issues, and falls, and these symptoms correlate with losses of acetylcholine in patients. Patients also have cognitive impairments such as poor attention. And the striatum not only uses primarily dopamine to drive movement, it also receives input from other systems that help shape movements. And Parkinson's patients, 60% of them fall, and those that fall tend to fall recurrently. So we've developed a circuit-based model in which we try to describe and explain why falls occur in Parkinson's patients. In the schematic here, there's cholinergic release of acetylcholine into the cortex, and this is critical for attentional processes. And this is important with movement in regards to detecting and acting on environmental cues, such as in humans when you're dealing with a distraction, when you're climbing stairs, when you're turning, you need to update your movement constantly and report that down to the striatum. And in the striatum, of course, which receives dopamine terminals, which are lost in the disease, there's an integration and an attentional motor integration in the striatum, and this drives more complex movement. And we believe it's a breakdown of this process that leads to falls in Parkinson's patients. And we've experimented with different ways with lesions and shutting off different systems to see to try to better understand the breakdown of this communication. And as I said before, currently I'm using optogenetics. So the primary behavioral endpoint of our experiments is falls. So we measure falls using the Michigan Complex Motor Control Task, which is a beam traversal apparatus in which rats are trained to walk back and forth across end stations, and they have to traverse over these narrow rods, either a straight rod or a zigzag rod. These rods are rotated, and animals really have to focus their attention as they're traversing. They have to pay close attention to their body positioning, their gait, and their speed to avoid falls on these beams. So this isn't something's wrong with the presentation here.
It's the wrong one. All right.
Disaster. Okay, well, I'll just go on. I had different videos. Okay. So we first used we first did lesion models in which we lesioned various systems that I mentioned before, to try to replicate some of the losses that are found in Parkinson's patients. On the left here, we measured falls with different types of lesions. The first type of lesions are in the blue graph here. These animals received partial bilateral lesions of the dorsomedial striatum with 6-hydroxydopamine. In a separate group, animals in the green here, they received bilateral lesions of saccharin, which kills cell bodies in the basal forebrain. And in these two groups of animals, there wasn't a significant increase in falls relative to shams, but in animals that had both of these lesion types, which we call DL animals, duals, there was a significant increase in falls. And importantly, falls on this task correlated with performance on a sustained attention task. And the poorer they performed, the more they fell. And importantly, in the green group also had poor attention on the sustained attention task, because that's what saccharin lesions are known to do. However, they didn't fall. So what that says is that it's the underlying loss of dopamine in the DL rats that causes the falls. So we believe there's an unmasking of these dopamine effects that comes when there's a loss of acetylcholine into the cortex. And this is kind of what's seen in human patients. They have an underlying loss of dopamine. They may be able to get around, but it's not until they suffer from a loss of acetylcholine and poor attention that's when they really start to fall. So that's the first part of our series of studies. The second part was using chemogenetics dreads to shut down the basal forebrain transiently. And we use sign and goal trackers for this part of the experiment. Sign trackers, as most of you probably know, are used as a model of addiction. They attribute incentive salience to reward cues, and they also perform poorly on attention tasks. That's sustained attention tasks that I mentioned before. They tend to suffer as the task goes on, and they have more fluctuating performance than goal trackers. And you could say that goal trackers are more of a cognitively, they use more cognitive goal-directed behavior in their movement, where sign trackers use more habitual movement. So we injected an inhibitory dread into the basal forebrain. These are just the subregions of the basal forebrain. And then we injected either CNO or vehicle. And CNO works on dreads work on a time scale of about a half hour to a couple hours. And we were able to increase falls. And these were mostly seen in goal trackers. So in goal trackers, there are more falls, more slips, and a slower traversal speed. And the reason why goal trackers may have been more effective when shutting down the basal forebrain was that goal trackers tend to use their cognition more when they're for their more goal-directed behavior. So they will be more affected with these inhibitory dreads, where sign trackers just kind of are always go, 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 more habitual, and so they weren't as effective. So the next step in this series of experiments is optogenetics. So optogenetics, of course, works on a much faster time scale as opposed to permanent lesions or with dreads over a half hour to a couple hours. We wanted to see if we could almost instantly cause falls by using optogenetics. So we used chat-3 positive rats, and they received bilateral 6-hydroxydopamine lesions of the striatum, or shams lesions. They also received microinjections of an inhibitory IC++ virus into the basal forebrain, or a control virus. And we used wireless LEDs. In our early experiments, we used just regular laser, and we had a lot of wires to deal with, and it was really difficult on the traversal beams. But wireless worked really good after a while once we got the hang of it. With 3 experimental groups: inhibitory virus in the basal forebrain plus striatal lesions, inhibitory virus plus sham lesions, or control virus in the basal forebrain and striatal lesions. For this experiment, we had two parts. Animals will receive inhibitory pulses twice per run, 1 second pulses at the beginning and at the middle of each traversal with the straight and zigzag rods, or we did continuous inhibition, which lasted throughout the duration of each traversal. And I'll show you videos of those. They work. So this is just our wireless setup here. LED cannula were inserted just above the basal forebrain, which emitted pulses into the basal forebrain, which is attached to a receiver and a custom-made clip on the rats. Just some histology showing the basal forebrain subregions and co-localization of virus expression with cholinergic neurons. Just below that, we see an example of striatal 6-hydroxydopamine lesion, TH stain, showing a loss of TH in the dorsal medial stratum. So animals that had both of these manipulations, this is early in our pilot experiments. When we turned the lasers on, they fell almost instantly within a second. So it seemed to be working, but we wanted to expand this to a much larger study and use wireless LED and have good control so we can verify that there weren't any non-specific effects of the lasers. So we did a large study with a kind of a grueling test sequence, 17 days. Animals were first trained on the plank surface, the stationary rod, then the rotating rod. Then they got 1 second inhibitory pulses during the straight rod runs. Then they were trained on the zigzag and got 1 second pulses on the zigzag. Then they went back to the straight rod and then the zigzag rod with its continuous pulses. And in total, each rat underwent 128 traversals, half on the zigzag, half on the straight rod. Half of those trials had the inhibitory pulses. And we can see the groups here. We use males and females. We also use sign and flow trackers. And we divided them evenly into the 3 groups, as we mentioned. And so here's an example of an animal that has the control virus that underwent traversals on the zigzag rod, and this animal gets the two 1 second pulses per run. This animal has just the control virus with a 6-hydroxydopamine lesion. Gets 1 pulse there. Gets 1 pulse there. So the animal pretty much has no problem going across the beam. No falls for that animal. The next animal has the inhibitory virus, and it's going to receive an inhibitory pulse throughout the duration of runs, and also has 6-hydroxydopamine lesions. And you can see almost immediately it has trouble traversing the rod. It's already falling and slipping. Falls again. And multiple falls by this animal. So this animal, so I scored a maximum of one fall per zigzag section. So this animal had a score of 2 falls for the entire run. So into the data. First, on the straight rod, there was not a significant group time stim interaction. On the left are the no stim runs, and then on the right are the effects with those 1 second LED pulses. And these are the 3 groups: the inhibitory virus plus the DMS lesion, dorsal medial striatal lesion, the inhibitory AAV plus sham lesions, and then control virus with striatal lesions. There was a little bit of a trend for increased falls in the red group, which is the inhibitory virus plus striatal lesions. And there was 21 out of 25 of them fell more with stim, but this didn't quite reach significance. I should note that the squares are goal trackers and the circles are sign trackers. We divided them up evenly, but for the sake of brevity here, I'm not gonna. We found some interactions, but we didn't have a clear conclusion on the difference between those. So these are just combined in this data. Next is looking at the straight rod with continuous LED pulses throughout the duration of the runs. Again, a little bit of a trend in the groups with the inhibitory virus. 18 out of the 20 in the inhibitory virus plus dorsal medial lesion group fell more with the stim, but there was no group time stim interaction. On the zigzag rod, however, there was pretty robust effects of the inhibitory pulses, both in the 1 second and the continuous. In the 1 second here, you see most of the animals, almost all the animals fell more often with the stim. We have 1 outlier there on the bottom that didn't fall at all. And this effect was not seen in the group with the inhibitory virus and sham striatal dopamine lesions, and or the control group with the control virus. And this is with a continuous pulse throughout the duration of runs. Throughout the duration of the runs, the same effect. More falls in our main experimental group, and no falls in the absence of the dopamine lesions and in the control groups. So that's it. Data's pretty clear cut. Just some summary. Falls increased with activation of the inhibitory ICC plus virus in the basal forebrain of Chat-Cree rats, Chat-Cree positive rats that also had 6-hydroxydopamine lesions. And this was fun. This was particularly more seen with the zigzag route, which is more cognitively demanding. And this was true for both the short pulses and pulses that lasted throughout the duration of the run. Activation of the inhibitory virus without the striatal lesions did not increase falls, and falls were also not elevated in rats with a control virus, but did have 6-hydroxy lesions. And these findings corroborate our dual lesion model that I showed at the beginning, in which loss of acetylcholine from the basal forebrain unmasks an underlying loss of dopamine. And that brings to the surface a heightened fall propensity, similar to Parkinson's patients and Parkinson's patients that have cognitive impairments and loss of acetylcholine. And a wireless LED alteration or enhancement of cholinergic output may be a useful therapeutic tool to improve movement deficits in patients with cognitive symptoms and high fall propensity. And that's it. This is the now non-existent starter lab. I'm now working to finish up my projects with at Barrett's lab. So thank you to Kent and thank you to Martin.
Okay. All right. Very good. Thank you, Aaron. So any questions for Aaron? That was very clear cut.
Yeah, I want to look quick.
Yeah. So, so Aaron, what about, so I think there's some M1 in there. I forget, are you following up on trying to do some M1 stimulation as a substitute for?
Yeah, we've done some, we have done some drug studies in the past, but right now, all my focus has been doing the optogenetic stuff. Just getting the experiment up. It's a big experiment, so that was my main focus for now.
No kidding. So excellent. Okay.
Okay. Yeah, I have a question here. So
Sure.
yeah, it's very beautiful work and congratulations. I'm not very familiar with this double lesion model, but very curious why we lesion dorsal medial part of stratum instead of dorsal lateral stratum.
So dorsal medial, of course, that's probably what in Parkinson's patients, that's where there's this motor, this attention motor interface where the dorsal medial stratum receives input from more of the cognitive areas of the cortex, and that and I guess it would kind of make sense to lesion the entire stratum, but there's we can't really do that bilaterally, or that animals won't be able to move at all. But so we wanted to focus on more the tension at the part of the stratum that gets more cognitive input from the cortex, because this is more of a cognitive motor model.
Mm-hmm. Yeah. So I just to underscore what Aaron said, I think the bigger lesions, the animals wouldn't perform very well. And what's impressive is Aaron and Martin get these very robust effects with these relatively restricted 6-hydroxydopamine lesions. So it's a really clever model that they've developed. So,
hey Aaron, could you flip back to the scatter plots?
Yep. Sure.
Yeah. So how big is? I mean, it's certainly smaller than it is for the virus plus lesion, but how big is that effect size for the inhibitory? Like, I'm wondering, do you have a small signal there?
The effect size for the inhibitory group?
Yeah.
Relative to, like, just in that group relative to stim versus no stim runs?
Yeah, or just, like the within subject Steve kind of
thing. I don't recall exactly what the value is, but it is pretty robust. Almost every animal had more falls except for one outlier. So it's a pretty big effect size.
Yeah, so I'm just wondering, like it may be going a little strong to say there's no effect for them, and maybe just saying that it's smaller, because I think I'd be interested to see what the Bayesian is on that.
Yeah, yeah, I don't have the exact data with me right now, but the
Okay,
stats
yeah,
with me right now, but
just
that's,
curious.
yeah,
Yeah, I mean,
yeah,
I mean, it would make sense if they have, just some cognitive aspects too. But
yeah,
all right, thanks.
thanks.
All right. Any other questions for Aaron? Okay. Well, thanks very much. I thank all the speakers today for some really fun talks. Much appreciated. And I think we're just going to have a short executive committee meeting now.